Good afternoon, everyone. So I'm Bruno Speder, and I'm the VP Regulatory Affairs and Consultancy at the Open Orphan Group. For those not knowing our company, very short introduction. So Open Orphan has two subsidiaries, hVIVO, which is our challenge unit and challenge excellence center based in Whitechapel in East London. And on the other side, you have Venn Life Science, which is our drug development consultancy group and our clinical operations group based in Breda in the Netherlands. Myself, I'm based in Antwerp in Belgium, and I divide my time over our London, Paris, and Breda offices. In our Paris office, we have our data management activities. So thank you all for joining this presentation today. The aim of this presentation is to guide you through the story of the regulatory approval challenges we had with the approval of the world's first SARS-CoV-2 challenge model. It was an exciting journey with quite some challenges, as you might imagine, and we would like to walk you through the story of this approval. We'll firstly quickly give an introduction on human challenge trials, for those not too familiar with the subject. Then we have some discussions on the SARS-CoV-2 challenge model. We'll walk you through the regulatory guidance available on the subject. Then, of course, we'll come to the UK Covid-19 challenge program, of which the characterization study we run was a part. And then we'll briefly guide you through the next steps of the model, specifically in the regulatory context. Now, what is a human challenge trial? A human challenge trial or a controlled human infection model, both are synonyms, are actually studies in which we make people ill on purpose. So the participants in this type of studies are challenged with an infectious disease organism, which we call a challenge agent, and then are administered a vaccine or an antiviral, depending which product you would like to test. This challenge organism can be very close to the wild type and pathogenic, but can also be an adapted or attenuated form with a lesser or no pathogenicity, and can also be genetically modified in some manner, a bit depending on the organism you would like to test. Now, these studies actually have a very long history. Actually, they started very early on in 1796 when Edward Jenner actually administered the first cowpox when testing the first cowpox inoculation, actually showing that the history of challenge studies and the history of vaccine development is very closely linked. In the 1900s, we had Walter Reed with his yellow fever experiments to support the search for a treatment for the US troops that were at that time deployed in Cuba. In 1932, we had the Tuskegee syphilis study where syphilis was administered to subjects in the southern United States without their knowledge, showing that these challenge studies have not always been used in the most ethical way. But they really had a breakthrough since the Second World War, in particular since 1947, when the UK government established a Common Cold Research Unit near Salisbury to actually try and find a cure for the common cold through human challenge studies. And in 2016, we had the Vaxchora cholera vaccine, and it was actually the first time that the vaccine for the efficacy part of the regulatory dossier was approved based on a human challenge study, and that vaccine has been both approved by the FDA and the European Medicines Agency. Today, the human challenge model is used in a very wide area of infectious diseases, going from influenza to cholera over RSV and malaria, but also in a number of more exotic indications, challenge models are available. So these trials actually have a long history and a broad range of applications. How does such a study look in practice? So these studies are usually performed on healthy volunteers, although in some specific cases, for example, they can also be used in asthmatic subjects, for example, or subjects with some underlying conditions, if there are particular research questions you would like to study in your clinical trial. And they're performed in specifically designed clinical facilities. And these are performed in quarantine units where the subjects are really in quarantine and isolated in isolation rooms. The reason for that is twofold. It's to avoid cross-contamination where subjects infect each other, but also to avoid outside viruses to infect subjects and to create noise in your clinical trial. We use a reversed barrier setting nursing to avoid contamination with very strict infection control measures. And actually, if you would look at the human challenge unit, it would be very similar to a phase 1-like setting. So for those familiar with phase 1 studies, a human challenge study quite looks like a phase 1 unit, but with these quarantine and isolation rooms. If you look at the study design aspects, characterization trial, so the trial where you first try out your challenge agent to find the right dose. This study is usually an open label design, single ascending dose with multiple dose ranges, very similar to a phase 1 study you would run with the drug, except that it's open label. And for vaccine and antiviral studies, this is usually a double-blind design, or in certain cases, an open label study could also be envisaged and discussed with regulators. How is the study flow of such a human challenge study? There's a little difference between vaccine studies and studies with antiviral, or if you go for a bacterial challenge agent, antibacterial challenge agents. In the vaccine study, you see two big phases. A non-quarantine phase in green on the slide, and a quarantine phase in blue on the slide. Subjects are screened, general health, inclusion-exclusion criteria, and in particular on their seronegative or seropositivity rate are administered the vaccine, and 4, 6, 8 weeks later, depending on the study design, they're admitted to quarantine, challenged with a challenge agent, have to remain in quarantine for a number of days. It goes from 3 days for malaria studies to 11 or even more days for an airborne pathogen. Then they are discharged, and then there is a follow-up phase. An antiviral study is, broadly speaking, relatively similar. Subjects are screened, but then they're administered in quarantine. They are then challenged, and then they get an antiviral product. And also there, they have to remain in quarantine for a number of days, the number of days depending on the organism, and then they are discharged and followed up. So that's how, broadly speaking, challenge studies look like. What is actually the added value of challenge studies in drug development? They have multiple uses and highly dependent a bit on the drug and the pathogen that are being studied. The most common use is the proof of concept, where a particular vaccine candidate or antiviral product is being tested to have a first indication if a product could work or not. But more and more, they have also been used as dose-finding studies. In example, EMA has accepted human challenge studies as a dose-finding element to replace a dose-finding field study in the past. But they could also be used in more fundamental scientific research to have a clearer understanding of the pathogenicity and the immune response to a certain organism, or to identify potential immune correlates of protection. In certain very specific cases, they could also be used to support the emergency use of a vaccine or an antiviral product, or as we have seen in the Vaxchora case, which was a cholera vaccine for travelers, used as the efficacy basis for licensure. In the case of SARS-CoV-2 and SARS-CoV-2 vaccine developments, also there is a broad range of applicable uses, as you can see in the table. But in the current state of the pandemic, a challenge study could be used for two specific elements. It could be used to test the shedding profile of a vaccine. As we see in the current status of the pandemic, we still see that the current vaccines do not offer full protection and block transmission. So a challenge study could be a very useful way to test specifically on the shedding profile of existing vaccines, but also on the shedding profile of vaccines in development, where they could offer an added protection or an added value for new vaccines, but also to be able to quickly test the efficacy of booster vaccines against variants of concern, and in general test the efficacy of booster vaccines and different booster vaccine regimens. Another option could also be in challenge studies to quickly test dosing regimens where different types of vaccines, for example, an mRNA and a non-mRNA vaccine are combined. If you look at their potential role in the development of antivirals for COVID-19, also there are different possibilities, but also here it could be used as a rapid determination of the efficacy of antivirals, where you can test the efficacy without the confounding factors of comorbidity and that you would see typically in a hospitalized setting. And the challenge model has been used quite broadly in the efficacy testing of antivirals, for example, in RSV or influenza. If you looked at the two tables before, we actually see that yes, there is a need for COVID-19 challenge studies, and they could help the development of COVID-19 vaccines and COVID-19 antiviral products. And in essence, the approach you would take for a SARS-CoV-2 challenge study would not be highly different than the one you would take for influenza or RSV, other airborne pathogens. Now, of course, influenza, RSV are well-described illnesses. We know how the disease progresses. And for SARS-CoV-2, of course, as we've seen the past 1.5 years, enhanced safety considerations are really necessary when running these studies. Two important points there are the potential serious consequences of the infection, acutely, but also on the longer term with long COVID, and also the lack of treatment. And there we also have to take into account that that the setup of the challenge model was started summer 2020, when no treatment was available yet. And these two elements actually give thought and say, okay, is it actually ethical to conduct SARS-CoV-2 challenge studies and to use this model in the development of vaccines and antivirals for COVID-19? Now, if you look at the regulatory guidance on human challenge trials, there are actually two sets of guidances that were available at the time when we started to set up this model. First of all, you have the general World Health Organization guidance on human challenge trials, which you see here on the slide. So the WHO has two guidances that discuss the use of challenge studies in development of vaccines. But the WHO also quite quickly, when the pandemic started, published guidance on the ethical acceptability of COVID-19 human challenge studies, but also on the feasibility and the more technical aspects these challenge studies should adhere to in the view of the World Health Organization. What in a very brief summary did the WHO advisory group recommend? They recommended a two-step strategy. First of all, that the challenge agent should be well characterized and studied in a dose escalation study. Before that, in the second step, vaccines and antiviral products could be tested in such an environment. That the volunteers should be restricted to very healthy individuals, and the WHO recommended 18 to 25 years of age. Studies to be performed in high-level isolation units. Also, they're quite similar to an influenza and RSV study. That there should be a legal quarantine, so that subjects should not be able to leave the legal quarantine, and we'll discuss that aspect also a bit later in the presentation. They gave some recommendations on the clades to be used. Also there, take into account that this was quite early in the pandemic. We didn't have all the variants that we know now. That the GMP manufacturing should be under BSL-3 capacity. WHO recommended a number of doses to be used in a characterization study. But the WHO didn't reach an agreement, is there a need for rescue treatment? And that's, as we will go through this presentation, has been one of the red lines and the topics of debate is it ethical and feasible to run SARS-CoV-2 challenge studies, knowing that there are potentially serious consequences of the disease without having a rescue treatment available? Now, how did the UK COVID-19 challenge program look like? This program was funded by the UK government through the UK Vaccine Task Force. And the Human Challenge Program, part of this UK Vaccine Task Force, was a partnership between Imperial College London, hVIVO, the UK Department of Business, Energy and Industrial Strategy, and the Royal Free London NHS Foundation Trust, where the study would eventually be run. It was supported by an extensive clinical advisory network with virology and vaccine and epidemiological experts from the University of Southampton, University of Oxford, University College London, University of Liverpool, the Wellcome Trust, which is very active and very supportive in human challenge studies, and the UK MHRA, so the UK Medicines and Health Regulatory Agency, the UK Drug and Vaccine Regulator. And the aim of this COVID-19 challenge program is to set up the COVID-19 challenge model. In the first phase, the SARS-CoV-2 characterization study. So in the second phase, antiviral and vaccine studies could be run, as well as more fundamental research studies. Now, if you look at the timelines in which this program was run, and it's for you to have an idea in which time frame this was run, the GMP manufacturing of the challenge agent started in September 2020. The non-clinical animal work was performed just in the new year. We got approval for the characterization study February 2021. First volunteers entered quarantine in March. Last volunteer left the unit in July 2021. And the full data of the study will be available just before the summer of next year. We already have a set of data, and we will go through the high-level results of the trial later in this presentation. But as there was an extremely extensive set of data and assessments done and collected, the full data analysis will take another few months. What's very important is that, and as we will go through the presentation, is that we, at each stage, we had to work with the data and the knowledge that was available at the time. Which means that at hindsight, we might have done certain things differently in view of the progressing scientific knowledge. But it's important to keep in mind that this program was done under accelerated timelines in a very dynamic and constantly changing environment, both scientifically, as ethically, as regulatory-wise. How did we, in practice, approach the regulatory side of things? We had extensive discussions with regulators. There is, of course, the manufacturing of the challenge agent, the non-clinical development, then very important, the study design and the safety, ethical discussions around it. From a regulatory perspective, also the extensive discussions we had on rescue therapy, where there were some regulatory hurdles in discussion, but also in availability. We had the trial approval process, both from the ethics committee view as from the competent authority review, and of course the quarantine facility itself. When we first set up the human challenge model, and when we initiated the first discussions, we initiated the first discussions with the European Medicines Agency. The reason for that is that it was the original aim, and it's to have a model that would be usable and applicable not only in the UK, but also in continental Europe. And in the view that summer 2020, Brexit was still not a fact and an ongoing process, we made the very early decision to have a rapid COVID-19 scientific advice with the European Medicines Agency, not only to discuss some practical aspects of the study, but also to have a first feel if the European Medicines Agency would be open to eventually consider data coming from a human challenge study in a regulatory review process. So we submitted a rapid COVID-19 scientific advice where we discussed the proposed manufacturing process, discussed the proposed non-clinical development plan, but also proposed the first high-level study design. And our aim was to see, okay, what would be the first feedback from a regulator on the idea of doing a COVID-19 challenge study. And we had a very rapid feedback from the European Medicines Agency, and that feedback was very supportive and positive. We had some technical discussions on the manufacturing process. The European Medicines Agency broadly agreed with our non-clinical development plan, gave favorable feedback on the study design, but also came back as you would expect with the feedback that, of course, the study design itself should be discussed with the national regulator, and of course the feedback from EMA there was indicative. EMA gave some very valuable feedback on the safety assessments, but also, most and utterly important, on the rescue therapy. At that moment, we had proposed as rescue therapy remdesivir, so Veklury from Gilead Sciences, which was at that time the only available rescue therapy. But with the complication, that product at the time did not have marketing authorization, neither in Europe as in the UK. So we had the regulatory complication to actually propose a rescue therapy that was not a commercially available product, which has some implications on the study design and the regulatory process. We also had a number of discussions with the UK regulator, the Medicines and Healthcare Products Regulatory Agency, on the manufacturing process, on the non-clinical development plan, but also on the study design, where there have been a number of major changes and discussions on that design. And during the course of half a year, we had multiple meetings with the MHRA, who was extremely flexible and extremely supportive in helping us getting this study through. And during the process, we of course had extensive advice and consultations with the clinical advisory group, composed of high-level virology and scientific experts from a number of UK universities, as we discussed in the previous slide. So just to illustrate that actually this was a very dynamic process where on the manufacturing and non-clinical aspects, we had quite an early agreement with the regulators on how this process should be developed. We had some very pragmatic feedback from both EMA as MHRA on certain GMP aspects, where both agencies were very supportive on a risk-based approach and on the acceptability that, seeing the accelerated nature of the program, full GMP and fully compliant with GMP was not possible, and that the scientific and regulatory sound approach was certainly acceptable. But most of the discussions were on the study design, where, of course, science and clinical knowledge changed dramatically over the 6 months that we ran this program, as we will discuss a bit later. We cannot discuss the regulatory aspects without discussing the ethics aspects. So the HRA, which is the UK Health Research Authority, and for those not familiar with the setup of ethics committees in the UK, is the overarching body governing ethics committees in the United Kingdom. Very quickly set up a specialist ad hoc research committee to evaluate COVID-19 challenge studies. This ethics committee was composed of a number of expert and lay members from UK Regs, and they were specifically selected on their experience in phase 1 healthy volunteer studies and in experience with vaccine studies. Their assessment was based on the WHO advisory guidelines, the ones we discussed a bit earlier, and they took a holistic view on the need for a SARS-CoV-2 challenge model. Indeed, the first study they looked at was the characterization study. But of course, you cannot approve a characterization study with having with the challenge model program in mind. Because not approving the characterization study would have meant that there would never have been a COVID-19 challenge model. So the ethics committee took a very holistic two-step view. What is the risk and the ethical consideration for the particular volunteer in view of the global program? And if you are interested in reading their assessments and their discussions in detail, there's an interesting article that the Ad hoc Research Ethics Committee published, and the reference is in the last slide, but it's also here. Also, with the Ethics Committee, we had a very practical and intense discussions during the setup of the model, where we also had very supportive and very transparent discussions, particularly on the setup and the design of the study. Looking at the manufacturing aspects, the strain that was selected was representative of the virus circulating in the UK over the summer of 2020, which was pre the Alpha, or previously known as the Kent variant. So the strain we selected was the original Wuhan strain, which was isolated from a patient with a suitable medical history and where suitable informed consent was in place. We had a number of strains selected, and this particular strain was chosen because it propagated well on the cell line, had no atypical growth characteristics, and presented no specific immunogenic changes. And also, it was free of adventitious agents because the patient we selected had very limited comorbidities, which was quite a challenge to get this particular strain. How was this manufacturing process run? In very high level, the manufacturing was performed under biosafety level 3 conditions. We had early discussions with regulators, both the MHRA as the Health and Safety Executive in the UK, which is responsible for biosafety, if BSL2 plus conditions would be acceptable, but there we got the feedback very early on that seeing the state of the pandemic and seeing the agent biosafety level 3 conditions were required. So the strain was manufactured under biosafety level 3 conditions. In a manufacturing process that is very similar to any other human challenge agent that would be manufactured in human cell lines, as you see here. For the non-clinical aspects, we had very early discussions on the suitability and the selection of animal model. And initially, we had 3 animal models that were considered: mice, ferret, and Syrian hamster. And the aim of this non-clinical study was to support more dose selection than safety, as we quickly realized that none of the proposed animal models would be very predictive of the safety aspects of the human challenge agent, but would be more appropriate to use for dose selection. Following discussion with both EMA as the MHRA, we selected the Syrian hamster as the animal model of choice, and the non-clinical characterization was performed in the Syrian hamster. When coming to the study design and the approach, that's, as you might imagine, the point where we had most of the discussions on. first major discussion point was which population can we select to be included in such a human challenge study. From the beginning, we choose the approach to only take the fittest of the fittest. Young, very healthy volunteers that have minimal risk of complications and minimal risk of long COVID. The age range originally proposed was between 18 and 25. There were extensive discussions on that, as a balance had to be made between taking very healthy volunteers to reduce the risk, but also to choose an age range that would be predictive and usable in later human challenge models. The risk was with only taking subjects from 18 to 25 years old is that we would have a model that was only tested in very healthy 18 to 25 years olds. So following discussion with the ethics committee, with regulators, finally the age range was between 18 and 30 years old to avoid actually only having 18, 19, 20 years old, very healthy subjects in the study, and that could potentially give an inappropriate and mixed view on the results and not be totally predictive for studies with vaccines and therapeutics. On the safety assessments, also there we had an extensive battery of tests, where we also selected and tested to be included subjects on a wide range of potential comorbidities. An intensive discussion we had with both the MHRA as with the ethics committee was on the Q-COVID questionnaire, a specific questionnaire developed to assess the risk for long COVID and for the risk of serious illness of SARS-CoV-2, where the MHRA and the ethics committee were not aligned on the need of that questionnaire, or if that questionnaire should be used as a very clear cutoff, or just to be used as a supportive instrument for the principal investigator to use in the selection of subjects. The safety assessments, as you might imagine, a very extensive battery of safety assessments was put in place where subjects were very, very closely monitored before they entered the study, but also during the study, and are still closely followed up in the post-study phase. The rescue therapy was a very debated aspect of the trial, both on the availability but also on the need. The time when we plan to run the study, the rescue therapy of choice would still have been remdesivir. But the problem with remdesivir as direct antiviral agent that it has to be given quite early in the infection cycle. And the initial thought of almost giving remdesivir preventively, specifically as it's a non-commercial product, giving additional regulatory hurdles as it would then have been considered an investigational medicinal product in the study, which would have made certain things a bit complicated, in particular in the setup of the study, led to, following extensive discussions with the MHRA and the ethics committee, not to as such give remdesivir preventively, but if a volunteer would be seen to pass certain thresholds in the study, virological but symptomatically and general safety, the subject would then be transferred to the intensive care unit, and then the intensive care staff would decide on the course of treatment. The aim of the discussion was also for, and that came specifically from the ethics committee, was that there was a clear feeling that if a subject would become ill outside certain parameters, it was more advisable for the health of the subject for them to be transferred to an intensive care unit than to be treated in the specific setting of a human challenge unit. This also led to the use of the Royal Free Hospital in London as the clinical trial site, seeing that it is a dedicated intensive care unit for infectious diseases, and the reference hospital in the UK for that effect. Recruitment and consenting process, although not something that was discussed with regulators, important in the approval of the study, is how do you recruit and consent subjects for a study where there was a real risk of serious illness, long-term consequences, and potentially death. So the recruitment process was extensively designed using the expertise of the ethics committee, external specialists, to come to a robust consenting process in multiple steps, where also the consenting process was recorded to make sure that subjects were following the consenting. Volunteer compensation, also there a touchy subject. For what exactly, which aspects can you compensate volunteers? We use the standard guidance on volunteer compensation in the UK, where we compensated the volunteers for the time in the quarantine unit, but also for the time they had to remain in isolation before entering the study, and for the time they had to spend in isolation and follow-up assets of the clinical trial. Now, finally, so how did the characterization study finally look like? So this is how the study looked like. So with the title, a dose-finding human experimental infection study in healthy subjects using a GMP-produced SARS-CoV-2 wild-type strain, SARS-CoV-2 characterization study. You can find the details on clinicaltrials.gov when searching on these parameters, with Professor Chris Chu from Imperial College as the chief investigator, supported by principal investigators with extensive experience in human challenge study. The challenge agent was administered intranasally. Study was run in healthy volunteers aged between 18 and 30 years inclusive. The trial was designed as a dose escalation study, single ascending dose, as you would expect, with 3 dose levels selected. The sentinel group was to be included at the start of each dose, and there was a safety review committee meeting after each 10 subjects. A lot of discussion was also on the frequency of the safety review committee, as we wanted a study that where safety was followed up very closely, but also without wanting to make the study very burdensome and not be able to move forward. So the safety review committee was held after each 10 subjects, and before each dose level, a sentinel group was foreseen. What were the endpoints of the characterization study? First of all, as in any characterization study, evaluate the safety of the wild-type SARS-CoV-2 challenge agent in the healthy subjects. And the second endpoint was to identify the inoculum dose that safely induced laboratory-confirmed infection in more than 50% of the participants. There was a very wide range of secondary endpoints. These are the primary endpoints. But if you look on clinicaltrials.gov, you will find the full details. The study was performed at the Royal Free Hospital in London, in Hampstead, and was performed in the BSL-4 quarantine unit there. There was no specific regulatory need to run it in a BSL-4 quarantine unit, but seeing the pandemic, and also seeing that the Royal Free is also a emergency hospital, their BSL-4 unit was the most suitable place to run the study. Also because it's very close to the infectious disease intensive care unit, in which the Royal Free Hospital in London is specialized. What was the outcome of this study? Here I might want to disappoint you, is that we can only give high-level results at this stage. We have more data available, but as you might appreciate, this was a study and a program run with government support, and there are we have some strict guidelines on what we can and cannot share on data at this moment. So these are the data we can share for the moment. A scientific publication is being written at the moment, and will be available, should be available soon. Should you be interested in receiving this publication, you will have my contact details at the last slide. So please do not hesitate to contact us. If you'd like to receive a copy of that publication, we will be happy to provide it to you. So the trial outcome was that 36 seronegative unvaccinated subjects have been safely inoculated. We had no serious adverse events in the study. We also, up till now, have no safety concerns in any subject, and no rescue treatment had to be required to be administered. The disease was being mild, as expected, with mild being seen as we had some subjects that were quite ill, but quite ill as you would have with a severe flu. But no one had complications or needed to be hospitalized. We had seen no changes in cardiac rhythm, which was something that was feared when we set up the model. But when we first wanted to initiate the model in September 2020, but based on the scientific and medical knowledge available at the time, we actually ran the study. Was less of a risk than we had anticipated initially. The challenge model is well tolerated. The challenge model is well tolerated, and the most common symptom and the most profound symptom next to the other expected symptoms is anosmia, loss of smell, and we also had quite some subjects with loss of taste. The current status is that the subjects are in the follow-up phase with some in-depth data analysis ongoing. As said, we cannot share at this moment any further data, but once the publication is available, we would be happy to share it with you on simple request. What's next? The model was set up in seronegative subjects, so where we selected specifically subjects that didn't have SARS-CoV-2 antibodies and were non-vaccinated. This model is now being converted to a seropositive model, particular for two reasons, is that a large part of the European population, age 12, depending from country to country, is now fully vaccinated. So the model is being converted to a seropositive model in a follow-up study, so that it can be used for later development in a vaccinated population. We've had further discussions with an MHRA scientific advisor to discuss the use of the model in an antiviral challenge study, where one of the main discussion points was with the MHRA was the safety assessments to be performed in the follow-up study. The characterization study had very extensive safety assessments, where up to the point where actually they were at the borderline of being practically feasible. Actually, we came to the conclusion that by implementing all the safety assessments required, the volunteers only came to 5 to 6 hours sleep per night, which is very burdensome on the volunteers, but also undermines the practicality of the model in efficacy studies. These safety assessments were very important in the initial setup of the model, and safety was very closely managed, as it should have been. But for next studies, now that we have more insight in the model, we will go to somewhat reduced safety assessment while still being very robust and solid. We're currently manufacturing a Delta virus challenge strain on behalf of Imperial College London, sponsored by the Wellcome Trust. The challenge studies are currently are being considered for the future development of new vaccines, but also of booster vaccine regimens, and to assess future treatment options. And currently, the University of Oxford is running a reinfection human challenge model where people previously infected are being reinfected with the Wuhan challenge strain. For those interested, there is some useful guidance available. Mainly the WHO guidance you see here. But there's also some useful literature for you to look through if you would be interested. Some of the articles we discussed, but also some other articles that could be useful for you. Should you want any of these articles, please also feel free to reach out to us, and we would be happy to provide them. This comes to the conclusion of the of this presentation. I see there were some questions through the chat, so I need to be able to find them. But also, if you have any further questions after the presentation, feel free to reach out to me, and we would be happy to present these to answer your question. We can go to the questions asked during the chat first. If your question has not been answered, feel free to send us an email, and we would be happy to answer them. Also, the presentation and the recording will be made available. So for those that are interested, you can rewatch. One of the questions I see here is: has Montelukast being considered as one of the rescue medicines? Yes and no, because at the time we were actually quite stuck with remdesivir for a number of reasons from the beginning of the study, which also not per se were linked to regulatory and ethical considerations, but also to logistic issues of availability. So it was considered, but for practical reasons, it was not included in the discussions. On the second question here, could you say some words on the number of trial participants from a statistical ethical point of view? 36 subjects were included. We had, based on the protocol, we could go further. Originally, we had anticipated to go up to 50 or 60 subjects. For practical reasons, we didn't go up. Once you have the article, it will be clear why we were able to have the study only run with 36 subjects, as the model in summary worked much better than we had originally anticipated. Question: will EMA and FDA produce guidelines similar to WHO? Yes and no. Both FDA and European Medicines Agency have indicated to us that, and also in the discussions myself and other colleagues had with both regulatory agencies on the usage of challenge studies, that they are certainly open to consider that these data, in part, as in the framework of an approval. Will they produce similar guidelines as WHO? My feeling is today not, as the WHO guidelines are quite overarching and robust, and have been referenced in our discussions with EMA. So probably they will not provide these guidelines. Another question I see is, this study was the first element of the UK government human challenge program. How confident are you that the program will pick up your findings and conduct subsequent human challenge trials on vaccine and antiviral products? If so, when do you anticipate these subsequent studies to commence, given the development of the pandemic? Very good question. Yes, we've had discussions with a very large number of interested parties, going from small biotech companies to very large pharma companies, on how a human challenge trial could fit in their development program. Some of the discussions are quite advanced. Of course, how can a human challenge study be run in the current context of the pandemic? It's true that there are a large number of vaccines. A number of vaccines, the ones you all know, are on the market. A number of treatment options have recently been approved. EMA very recently approved 2 treatment options. And one could say, okay, is there still use for this human challenge model? And our conviction is yes. And we also supported on that in discussions we've had up to, 2 hours ago with interested parties, is that what we see that this model is very quickly moving towards a model in a similar framework as we have for influenza or RSV. We have a fully vaccinated population, which makes it very difficult to find unvaccinated people, unless you go to countries where the vaccination rate is less. But it also provides an advantage, is that the challenge model now is, as you've seen in the early slides of the presentation, an ideal model to quickly test certain hypotheses. Does a particular vaccine work well as a booster vaccine? Challenge model is ideally suited for that. Does a certain vaccine or certain platform of vaccines reduce transmissibility of the virus? A human challenge study is a perfect model for that. So the model at this stage is perfectly suited to answer these questions and to quickly answer questions for the 2nd and 3rd generation of vaccines that are currently under development, to answer very specific questions. For example, does it work as a booster vaccine, or what is the relative efficacy compared to an mRNA vaccine or one of the other vaccines on the market? How was the consistency between each batch of the virus assured? Very good question. Actually, it was a problem that was not applicable to us, as we've only manufactured 1 batch of the virus, which might sound a bit weird, but actually is standard practice when manufacturing a human challenge agent. Is you manufacture 1 batch, which is very extensive. We can, I don't know the exact numbers by heart, but we can run a very large amount of challenge studies with that 1 batch. By the time all these studies would be run, the stock would not be relevant anymore. So we manufactured only 1 batch of the virus. Other than the UK government program, have you had contact with other government and commercial organizations? We've had some discussions with other government organizations, and we had a large number of discussions with different commercial entities or semi-commercial entities. Other question I have here on the chat is, when will the characterization study paper get published? The paper is currently under review. As you might imagine, this was a very complex setup with many parties, a large array of academic institutions, the UK government, UK vaccine task force. So that process is not running as quickly as we would want want it to be, we would be very happy to share a publication. So I can't give you a definite date on the characterization study, but if you contact us, we would be happy to share the paper once it's available to us, and then we would be happy to share it with you immediately. But we thank you for your patience. It's that we're working in a very complex environment with many people looking over our shoulder, many mothers-in-law, as one of my colleagues once joked, which make that process a bit lengthier than we would have hoped for. I don't know if there are any other questions at this stage. No, apparently not, which makes us exactly 4 o'clock here in London. As said, if you have any other questions popping up, feel free to contact us. You have my details here, and we would be happy to answer any questions you have. We hope you enjoyed the presentation. It was a very nutshell presentation of the very exciting journey we had setting up this model. I personally enjoyed it a lot. It was a very interesting and very intensive communication with regulators, ethics committee. As with any program, we learned a lot. Also things on hindsight we would have done slightly differently. We've also had quite a number of lessons learned, both from our side as from the ethics committee side, but also from the regulatory side on how other, for future pandemics, let's hope they're still far away, but how for future pandemics such a model could be set up quicker and more efficiently. So I thank you all for your attention. I thank you all for the questions. Hope it was interesting. As said, this presentation has been recorded. The recording will be made available to you, as is the presentation. And in case of questions, do not hesitate to reach out to us anytime. Thank you very much, and have a lovely day.